Okay, today we're going to add some more organisms to our ocean, the zooplankton. That is, those animal-like organisms that, well, they have many roles in the ocean, as we'll see. We'll first concentrate on their role in eating phytoplankton, but they have other roles as well. So what you're looking at here is a one way of catching some of the larger zooplankton. It's called a mock nest, and I'll say a few more words about that in a minute. You can see this is connecting up the net system to the boat, and here are the various nets. It's a way of deploying several nets at the same time and catching zooplankton at various depths. But before we get to that, I did want to say a few more words about the iron hypothesis. You remember it's the idea about how phytoplankton are limited in certain parts of the world's oceans, principally the North Subarctic Pacific, the Equatorial Pacific, and the Southern Ocean. And in these regions, there's very high levels of the other major nutrients, nitrate and phosphate and silicate. And it's been hypothesized and pretty much accepted now that primary production is limited by iron in these waters. All three of those areas are far from land, so that means they're not getting enough iron from land. And I mentioned on Thursday that another property of iron that makes it difficult for organisms to get at is the fact that it's insoluble at the normal pH of seawater, which is around 8 or so, and also when there's oxygen present. Different chemistry when there's lower pH and no oxygen, but of course in the surface ocean, it's lots of oxygen, enough oxygen in pH of 8. So it forms these iron oxides under those conditions and makes it difficult for phytoplankton to get at. As I mentioned, one piece of evidence for the iron hypothesis is these in situ iron fertilization. So they drove around the ocean in ships and dumped iron into the water and followed the system over time to see what happened. And of course, what they saw was the increase in phytoplankton biomass, primary production, and so on. So these places have been, this type of experiment has been done in several places, including the subarctic Pacific. The first one was actually done in the Equatorial Pacific, and then some of the more recent ones were done in the Southern Ocean. I think perhaps the most recent one was led by a friend of mine, Victor Schmedacek, who's a very interesting character. He's half Indian, Indian in Indian, and half German. So Victor, shows his German part, and his accent's kind of like a more of a kind of a mix between German and Indian. I met him in Goa, which is where there's a National Institute of Oceanography for India. Anyway, he led one of the latest ones in the Southern Ocean. So in other places in the ocean, though, that are not like the three HNLC regions, for the most part, it's nitrogen that limits primary production. And we know that from bottle experiments, similar to the ones that have been done with iron, showing the limitation of phytoplankton production by iron. That is, very simply, you add nitrate or ammonium, or phosphorus or iron, and you just see what happens with the phytoplankton to those bottles. And generally what happens is that in everywhere else but the HNLC regions, phytoplankton biomass increases when you add nitrogen, that is nitrate or ammonium, but not when you add the other nutrients. It's pretty much impossible to try to do an in situ iron fertilization because you need so much nitrogen to do that experiment. So that type of in situ fertilization experiment has not been done by oceanographers, although you could argue we are doing it as a society when we dump our fertilizer into, say, the Mississippi River. The Mississippi River flows into the Gulf of Mexico, and that creates phytoplankton blooms that would not be found there if it were not for the pollution by the nutrients that we add to the Mississippi River. Now, another piece of evidence that indicates that nitrogen limits primary production, or is the most limiting nutrient, is the observation that nitrate disappears before phosphorus. And this was this very crucial observation was made by Alfred Redfield back. You can see there. The paper was published in the early 60s, I believe. And Redfield, what he did was compared the concentrations of phosphorus and nitrate in various parts of the oceans. And you can see here that if you go back near where it's 0, basically there are many parts of the oceans that there's no measurable nitrate. So that is, nitrate is 0 here, but there's still measurable levels of phosphorus. And so the reason why that's the case is because of a process called denitrification. We're not going to get into that, but it's a microbial-driven process where nitrate is reduced all the way to nitrogen gas or another gas, N2O, and that goes back into the atmosphere. There's nothing analogous to that with phosphorus. So the oceans become limited by nitrogen because of that process, whereas they don't become limited, generally speaking, by phosphorus. Now, another very interesting observation from this graph is that there pretty much is a straight line between nitrogen and phosphorus, and that is the N to P ratio is pretty constant for many parts of the oceans, and that's been called the Redfield ratio. And it's basically, you can see that the numbers are 106 to 16 to 1. That's the number of carbon atoms to nitrogen atoms to phosphorus atoms. And that's worthwhile remembering, 106, 16, and 1. And you can see here that again shows that you need a lot more nitrogen than you do phosphorus. We're speaking generally for all organisms. This is not just marine organisms, but generally all organisms need more nitrogen because of the amount of protein that we have that's very nitrogen rich. We need phosphorus for lipids and nucleic acids. But those compounds, at least the nucleic acids, also need nitrogen. So we need much more nitrogen than phosphorus. And that observation that there's this kind of fairly standard or uniform ratio, that it doesn't vary a lot. And when it does vary, it's really interesting. But as a first approximation, it's 106 to 16 to 1. So that was one of Redfield's seminal contributions to the field. So we've been talking generally about what controls phytoplankton biomass and rates of primary production, and you should be able to list these properties. I should have gotten you to think about them before flashing up the answer. But the answer, of course, is light, nitrate, phosphorus, phosphate, and you should know which organisms require silicate, which of course means the diatoms, which are indicated there. And then finally, we have compounds like iron, which are, iron is only important in the HNLC regions. I mean, it's important everywhere. We always need iron, but it's only in the HNLC regions that iron is limiting. So we talk about and refer to all these factors as bottom-up factors, and that is that they affect the growth rate of the organisms. So what would happen if that was the only answer to the question about what controls phytoplankton growth in the oceans? What would happen if only bottom-up factors were operating? Well, we can kind of think about that and think about if we just allow phytoplankton to grow. And basically, plankton, phytoplankton, and many other microbes grow basically asexually. They may have a sexual phase to them, but for the most part, it's asexual. So they basically bud off and grow by division or fission. So one cell gives rise to 2, 2 gives rise to 4, and so on. And so that process is an exponential one. And so the equation that describes that exponential change in cell numbers over time is given right here. And this is worthwhile remembering. Very basic equation describing how growth changes over time. And it's basically just kind of very similar to the exponential decrease in light attenuation or light intensity in the water column. The only key difference is the fact that in attenuation equation we have a negative sign here, whereas in the exponential growth equation we have a positive sign. So when we graph that on a log scale, and in particular the natural log, although any log would work, we would get a straight line. And so we can see here that this organism is growing faster than this one over time. Okay, so we need that because now we can ask the question, okay, so to back up, I need to define my terms here. Mu is, again, I mentioned this before, is the symbol that's used for growth rate. That is the per capita change in abundance. N sub t is the number of cells at any time t, and so N sub 0 is the number of cells at time 0. So the growth at any time t is a function of just the initial number of cells times the e raised to the mu T, where T is time, of course. So with that, we could calculate how fast the oceans would actually fill up with phytoplankton, starting with one single cell. And for this little experiment, this little thought experiment, we're going to do two things. First of all, we're going to allow the oceans to be totally lit up, and so there's light going down way to the bottom. We're not going to worry about nutrient limitation. Just how fast would it fill up if you had just this tiny little cell that you would need a microscope to see. And also assuming a kind of typical growth rate for phytoplankton. Remember I said they double about every day. That means their growth rate is this, and you can see what the generation time would be given that growth rate. And so the answer would be, well, these are the possibilities. And I think it would be a useful experiment thing for you to do to figure out how long it would take. And just to give you a hint of the answer, it's not the longest time frame that we're talking about here. It's one of the two shorter ones. But I think it'd be worthwhile for you to figure that out on your own. The only other thing you need to know is find the volume for the oceans, which you can Google someplace. Okay, so the point is that even if it were 78 years, the oceans would quickly become just one solid plankton, mass of plankton, phytoplankton, if it were not for something else that got rid of those organisms. Or another way to say this is, or look at the problem is, what happens to the spring bloom? So you know why there's this increase. And we talked a little bit about the decrease or the lack of nutrients in the summer, but why would there be a decrease in the biomass? Perhaps you'd think, well, just the lack of nutrients would contribute to that decline, but why would they necessarily lyse and explode and disappear? It's not clear that the lack of nutrients per se would cause that to happen. So in fact, there's several ways in which you can get rid of the bloom and kill off phytoplankton. One is they can die themselves. I mean, so yes, the lack of nutrients could lead to cells themselves committing hara-kiri, so to speak. But another mechanism in the molecular parts of the story are link up viruses and programmed cell death, which I'm not going to get into. But suffice to say, there are viruses that basically hit all organisms, and we're going to talk a little bit more about those next week. But some are known to kill off phytoplankton. So one reason phytoplankton blooms stop is because of viral lysis. They could sink all the euphotic zone. Some of the phytoplankton, especially the bigger ones, diatoms, coccolithophorids, are big enough to sink, and so those could just disappear for that reason. And then finally, we get to the answer that we're going to be talking about for the rest of this lecture, is grazing by zooplankton. So that process of controlling the biomass of these organisms by, for the most part, grazing and viral lysis, we would call top-down controls. And it controls basically the biomass, whereas again, the bottom-up controls are things like nutrients and light. Now, this is a bit oversimplification because there are some complications and interactions between these, how lysis affects growth rates, for example. But as a first cut, as a way of thinking about these terms and a way of thinking about the factors, this is a good starting point. Bottom up, things like nutrient and light. Top down, processes like grazing and viral lysis. So we're going to be talking about zooplankton today and talk about viruses next week. So now the question is, how do we divide up this zooplankton community? It's somewhat analogous to the problem we face in talking about phytoplankton. So as with phytoplankton, size matters. And here's really roughly the three size classes that we'll be talking about. And today we're going to be focusing on the largest group of organisms, the mesozooplankton, and save the proteus for actually next week. And we'll talk a little bit more about the microzooplankton on Thursday. Another way to divide them up is their lifestyle. There are organisms that are always in the plankton, that is the holoplankton, and there are other ones where only their juvenile stages in the water column. And then we're going to end up today with talking about just the diversity of organisms that we see in the zooplankton. and leave for Thursday differences among feeding mechanisms. So what about size? Well, as with the phytoplankton, size matters. Now you should know already about why there's different types of size of different phytoplankton in the oceans. One answer is nutrient concentrations. That is, low concentrations favor small phytoplankton. Grazing, we'll see, comes into play too. But if we just think for a minute that there are in fact different size phytoplankton, that would immediately kind of suggest, I hope, that there need to be different zooplankton that graze on these different size phytoplankton. And the size range in the zooplankton could range from actually even less than 9 microns. They can be as small as a couple microns in size, all the way up to almost, well, certainly macroscopic that you can see without a microscope, millimeter size. Still not, whale size, blue whale size, but still fairly big when we talk about these organisms. And so because of the differences in the size of these organisms, there's different predator-prey relationships. So this is a graph showing just the predator size ranging from huge range in size, going from millimeters or even less than a millimeter up to blue whale size versus what they can eat. And we'll talk later about this graph and about the exceptions, about things like why salps and blue whales fall off the graph. But the point here right now, we're just concentrating on a simple line. That is, very simply, bigger organisms eat bigger other organisms. Or if you're a bigger prey, you are going to be eaten by a bigger predator. So that's the basic message here, very simple. And very roughly, that relationship is on the order of 10. Now, there are many exceptions, and I know some of my zooplankton colleagues don't like even talking about one number like 10, but I'm a big believer in having a couple things to kind of hang your hat on and organize your thoughts about. And I think this is a useful way to think about the size relationships between zooplankton and predators. So what that basically means is that we need, because there's different prey sizes in the phytoplankton community, we need different predators to deal with those different sized preys, because they're not going to be, one predator is not going to be able to eat all those different types of prey. And to attach some a bit more specific names to these size ranges, we can think about Prochlorococcus, which is one of those groups of organisms that I asked you to remember their name. It's a group of cyanobacteria, really small, the smallest, in fact, phytoplankton out there in the oceans, found in the lecithotrophic open oceans. And then diatoms are just the opposite. Could be 50 microns or 100 microns in size or so. And so just to give away a little bit of the answer from Thursday's lecture, it's the proteus that eat these smallest organisms, whereas these mesozooplankton, if there's any zooplankton that's going to eat them, it's going to be them. Any organism that's going to be eating the diatom is going to be the mesozooplankton. So that's why we need size. That's why we need size. But in fact, the mesozooplankton have more roles than just eating plants. That is, they have more roles than just herbivory. They do much more than just eat other plants. They also eat other grazers. So they're carnivores as well as being herbivores. And they may eat a ciliate, which in turn would be eating the phytoplankton. So they're a carnivore. The mesozooplankton are also fish food. We'll see in a moment that there are many types of mesozooplankton. They're great sources of protein for fish. So they're really important in that regard as well. And finally, their poop contributes to carbon export. Their fecal material, and this is a picture of a fecal pellet that's probably made by a zooplankton, mesozooplankton, a copepod specifically. And that sinks. Those are fairly big. 50 microns is pretty big in this world here. So that thing sinks down out of the water column and is part of the carbon pump that we talked about, the biology of carbon pump. That takes CO2, the phytoplankton take the CO2 from the atmosphere, the phytoplankton make this organic material, and then eventually sinks down to the deeper parts of the ocean and eventually to the sediments. And one way that organic carbon can sink is via the fecal material that's excreted by the zooplankton, and in particular the mesozooplankton, make poop big enough to sink out of the water column. So that's a non-trivial role of the mesozooplankton. Okay, so let's look a little bit about how we study these organisms. There's roughly 3 ways in which this is done. The classical way, the traditional way, is by nets. And then, of course, you can look at these organisms microscopically, or some of them are big enough that you don't have to look at them with a microscope. And a huge advantage of collecting them by nets over these 2 methods is that you can do controlled experiments. As I look at this, I realize that there's still another way that's only used with some very delicate zooplankton, the salps in particular, is by scuba diving, where people literally will jump in the water and creep up behind some of these organisms and catch them with a jar. I was on a cruise in the subarctic Pacific where Larry Maiden from Hui, Woods Hole Oceanographic Institute, would do that, and the water was incredibly cold and miserable conditions. But I remember him coming back up all red in the face because of the cold water. But he had these beautiful, beautiful animals in these jars, the salps. And I'm going to show you some pictures of salps near the end of this lecture. The other methods don't collect the material at all, but look at them more remotely, and we'll get into those in a minute. So here's different types of nets. There's many different types of nets. Here's the mock nest that I mentioned already, where you have it's a very complicated system of several nets that are towed together, and then you can close and open the nets at various depths or time or whatever you like to do, and you can collect many samples that way at once. This is what's called a bongo nets. You can see that has the 2 bongo-like shapes, I guess, to it. Advantage of having the 2 is just be able to collect that much more material at once. I'll try to remember to bring a plankton net. If you haven't seen one, it's worthwhile take a look at it. I won't bring a mock nest or anything this big, something much smaller that can be used to collect zooplankton. And here's what you get. Here's something called from a mock nest. So up here is some krill. That's a euphausid. I'll talk a bit more about that in a minute. Here's a salp that I mentioned already. And I'll show a much better picture of that in a minute as well. Kind of amazed that they were able to catch salp, because salps, as I mentioned, the reason why Larry had to jump in the water is because they're often damaged when they're collected by nets, and then they're caught. So if you could look at this, the sample ends up in this what's called the cod end, and here it's the same principle with the bongo nets. And they're all squished together, and sometimes they come up not looking so good. Okay, so that's by nets. The other way is by acoustic methods. Here you're not collecting the material at all. And basically this is really good for counting the number of organisms. And you may or may not get much information about what's there. And you're probably not going to get all the zooplankton, but the advantage of this is you'll get a really precise estimate of the numbers of organisms. And it's used by physical oceanographers to look at currents. So it's called the acoustic Doppler current profiler, or ADCP for short. And it's basically sonar. So what happens is that sound is bounced off into the water, and that sound hits the particles, which often are the zooplankton, and that sound is transmitted back to the receiver, and the oceanographers can deduce then how currents move, and then also the zooplankton ecologists can count the number of zooplankton they may have in the sample. Okay, and then the final method is called FlowCAM, and basically what it is, it's a computer, a flow cytometer, which we kind of talked about before, hooked up to a microscope. So so what happens is that the plankton are passed through and underneath a microscope lens, basically, and then pictures are taken. You can kind of see here on the screen various types of pictures of various types of phytoplankton, not phytoplankton, but zooplankton that were sampled by this by this instrument. This is a colleague of mine, Barry Scher, who's now retired from the Oregon State University, where Charlie Miller and Pat Wheeler are the authors of one of the books I recommend that you take a look at. And so here's what you get. You get these pictures of different types of zooplankton. This looks like probably maybe a crab, nopuli, copepod, and so on. So now it wouldn't help too much if you had to do this by hand and sit underneath the microscope or look at the computer all day. So what's done though is that these images are digitized and the computer actually does the counting of different types of organisms. So that's the advantage of this method over, say, the acoustic, the ADCP method, is that you actually get some more information, much more information about the types of organisms that are present. And you can perhaps also look at some of the larger phytoplankton. Professor John Cohen, who some of you may know, down here in Lewis, uses something like this to look at zooplankton, and he's using that method in the Delaware estuary. Okay, so we talked about size, just went over some of the ways we can study some of the larger zooplankton, the mesozooplankton, by nets, by acoustic methods, also by FlowCAM. There are other ways in which we can divide up the community, and that is by lifestyle. And the two basic divisions there are the holoplankton, which are always found in the water column, their whole life cycle is there, versus the meroplankton, which, as you can see there, only their juvenile stage is in the water column. So let's talk a little about the meroplankton. So, well, let's talk about the holoplankton first. So again, they're only found in the water column. We're going to talk more about these in a minute when I give some examples of various types of zooplankton. So the best example of a holoplankton is copepod. And this is a word that I want you to know. It's the most important type of zooplankton in the oceans. And I'll have a lot more to say about copepods in a minute. So holoplankton always found in the pelagic zone. They don't have it. They're adult. This is an adult. This is a female with eggs. That's what you're looking at here, the egg sacs. And there's just another picture of another type of copepod. Calanus is a genus of copepod. I want you to remember that because that is an important genus of copepods, worthwhile remembering. This just happens to be my favorite species, which, even though it's my favorite, you don't have to remember that. Okay, so in contrast to the holoplankton, we have the meroplankton. So meroplankton, again, the adults are on the benthic, in the benthic community. They're on the bottom. We're on rocks on shore, in the case of some. But the juvenile stages, the younger stages, are in the pelagic zone. And a good example of this is crab larvae. So crabs, you know crabs, they crawl around on the bottom, but their larval stages, their younger stages, swim, and they kind of look like this. Don't look much like a crab at all at this point. So we can further divide the meroplankton into two types, whether or not they feed. And the ones that feed are called planktotrophic larvae. So planktotrophic, the food is the plankton. So these organisms have to feed in order to survive. And the other type, so they feed on the plankton. And the other type is lecithotrophic larvae. So basically they carry a little lunch pail with them. When they hatch out from their mother, they come with some yolk from that egg, and they do not feed at all. And so they can't carry a big enough lunch pail to allow them to survive long in the pelagic zone. So they have to go back to the benthic or shore or the rocks or the bottom soon after they are hatched. And we'll talk later about some of the advantages of having this lifestyle, but you can think about that and think and guess about why there's advantages. Basically dispersal, about spreading your offspring into other potentially new habitats. Whereas the plankton, the advantage of being able to feed is that you can survive longer in the plankton. Now the disadvantage is you have to find that feed. So they have different roles in the water column. You can think about which types of roles both of these play, and that may be a good test question for you to think about. Okay, some examples. let's just talk about the lecithotrophic larvae. You can see here the jellyfish, polychaetes, gastropods, bivalves, and so on. Examples of meroplankton animals that have planktotrophic larvae. Again, you can see the answers here. Gastropods, bivalves, crustaceans, and echinoderms. So these are just some pretty pictures of some echinoderms. Okay, so we've talked about it already now. This should be meso, not macro rather. Some folks talk about macro, but I've gone to using meso, which is what Miller and Wheeler use. Basically the same idea. So we talked about the importance of size and divide up the zooplankton community. We talked about holoplankton versus meroplankton. And now we're going to talk about dividing these organisms up by taxonomy. And so again, as we saw with the phytoplankton, there's some advantages and disadvantages in talking about the taxonomy. I have to say I'm not a real big fan of dividing things up by taxonomy. I don't know if I mentioned this already. It's one of the things I don't like about biology is the taxonomic names. And I almost left biology because in the zoology course I took, we were told to memorize all these names, and I didn't like that at all. It becomes a little more interesting when you start to connect up the names with their function in the environment, and the names become a convenient shorthand for looking at that function, and also for looking at the evolutionary relationships among organisms. So, there is value in looking at taxonomic divisions and look at these organisms divided up by their taxonomy, by different types of where they are in the tree of life. So, what I'm going to do now is just give you some highlights of some of the zooplankton. And if nothing else, I hope you will appreciate the diversity and beauty of many of these organisms. So this is by no means an exhaustive list of all these zooplankton that we can see in the oceans. We're just going to hit some highlights and some that I like, some of my personal favorites. Okay, so we're going to start off with some Cnidarians. And so Cnidarians, if you are interested in corals, you would know about this phylum of organisms. There's the pronunciation. I had to look it up myself. You don't pronounce the C. So you can see some of the characteristics of these organisms. A pretty diverse phylum of organisms. We'll talk about corals later in the course, but with regard to zooplankton, we can talk about the jellyfish and other organisms that look like this and certainly have a role in the plankton. So as I mentioned, we're just kind of streaming through the tree of life here and picking up some of the important zooplankton, and some I have to say are just pretty and interesting. So ctenophores, sometimes called comb jellies. You can see the characteristics of these organisms. They're all holoplankton as opposed to the Cnidarians, which are not necessarily, although many of these jellyfish are, of course. The comb jellies are holoplankton. That is, they're always in the plankton. Now we're going to move to mollusks. Just one, skipping around a lot, of course, and in that we're missing out some of the gastropods. But we'll go to the mollusks. And one example from the mollusks are the pteropods. These can be several, a little bit less than a millimeter to several millimeters in size. So you could see these with your naked eye without a microscope. They again are always on the plankton. Moving up to annelids, that is the worms. Worthwhile mentioning annelids and especially polychaetes because some of these organisms have again a zooplankton like meroplankton life stage in their life cycle, but polychaetes are much more important than benthos, and we're going to come across that word, and so it's something for you to remember. Though we'll see it again in the middle third of the course when we talk about the benthic communities. So some of these organisms, some of the annelids can be holoplankton. You think about worms as crawling around in soil and in sediments, but some are holoplankton. That is, they're always found in the plankton. and some are meroplankton. Continuing our tour, our grand tour, come to the arthropods, and this is the most diverse and largest number of known zooplankton species are in the Crustacea class in particular. And many of these organisms can be both, are either holoplankton or meroplankton. I don't want to give the impression that an organism could be both. One single species is one or the other. It's not as if they switch somehow. It wouldn't make any sense at all. So if you're a holoplankton organism, that's what you are. If you're meroplankton, that's what you are. You don't switch from one to the other. But of course, the meroplankton organisms, by definition, have their juvenile stage in the water column, and so that makes them meroplankton, even though the adult is on the bottom. So the crustaceans include many organisms that you already know, crabs. We'll see again in a minute. Also barnacles, you may not know, is a crustacean, not a mollusk. It's a crustacean. So barnacles, there you go. Barnacles are filter feeders. They're on rocks, and they have a larvae stage that's in the zooplankton. Mysids, they're not like the barnacles. They're holoplankton. Worthwhile knowing because they're important food source for fish and whales. Krill, their technical name, scientific name is euphausids. So they are definitely holoplankton. Some can be bioluminescent, but they're famous because of they're a food source for whales and penguins. And also, I think you can go to a drugstore and get krill as a protein supplement. And maybe we'll have a sample of the krill. In fact, I don't know if I bought it last year or not, but I'm definitely going to buy it this year for all of us to try out. So anyway, that's krill. Definitely important for thinking about, especially the Antarctic food webs. Crabs. Crabs is a meroplankton. Of course, you know crabs. Their adult stage is on land, or on the shore among rocks. If it's not on your dinner plate. But it has this larval stages that are definitely in the plankton, so they're meroplankton. And we'll talk more about crabs and their life cycle later when we talk about the estuaries. Now we get to the real and most important group of organisms. Important because of their abundance and biomass in the zooplankton community. Also, we know the most about these organisms. They're the most studied group of zooplankton. So here's a copepod again with its egg sacs there. So why are copepods so abundant? And it comes back to their body shape. So they have this like torpedo-like body shape, which makes them really good at not only detecting predators but also escaping predators. This is all taken from this review paper. Published by this Danish guy a couple of years ago now. And so they have, again, they're really powerful swimming machines for their size. The other reason is that they can detect prey. They can detect it sometimes by chemical methods, also by the vibrations in the water that's caused by a prey that may swim by it. And then finally, finally they're able to find mates. So copepods have a little bit more interesting personal life than some of the other organisms we've come across. They do have rituals in terms of finding mates and actually engage in sex as opposed to the asexual modes of reproduction that we've seen so far. So they're able to do that well. And if you're able to do that well, you're going to pass your genes on to the next generation more efficiently than someone who's just a wallflower and stays in his or her own room and doesn't get out and doesn't find a mate. These copepods are not that. So that's another reason why they're so abundant and successful. Okay, then finally we have two more examples to go through, and that's the Chetonasts, one of my personal favorites. They're important not only because I like them, I just like the looks of this kind of evil looking thing. Can be millimeters in size again, but they're important too, not just because I say I like their looks. They're carnivorous, so they're eating other zooplankton. They themselves are good food source for fish. Again, they're holoplankton, so they're totally in the water column. And then their common name is airworms. So I think it's worthwhile for you to know that word Cnidarians. Just realized I'm not sure if that's spelled correctly. I'll double check. I doubt the O is supposed to be there. Okay, and then one last example is the close to us. So we're up in this part of the tree of life with the chordates, and that actually is the hemichordates. It has it with the chordates, but I'm pretty sure it's the hemichordates. Include the salps. So the salps are these kind of they look kind of like jellyfish, but they're totally different phylum, not related at all. They just kind of have kind of a superficial resemblance to jellyfish. You can see these are huge. These are size bigger than the swimmer there. The other ones that I told you about that Larry Maiden was catching in a jar were just, they're big. They're, several centimeters in size, about the size of your palm, not the size of a person's body. So they can be really important in terms of the feeding on other members of the plankton. So that kind of wraps it up. And you probably wonder, what the heck should I know besides maybe knowing which ones I like the best? Well, I hope you're impressed by the diversity of organisms, not only by their beauty, but by the fact that there's many of them. And nearly all phyla of life are in the plankton. We talked about this before, that one reason to look at the oceans is because they have representatives of nearly every phylum that we see in the biosphere. The other important point that you should take away from this is that it includes examples from both the holoplankton and the meroplankton. Again, holoplankton are always found in the plankton in the pelagic zone, whereas the meroplankton only have a juvenile stage in the pelagic zone, and their adult is on the bottom. So again, once a species that's holoplankton, that's what it is. And a species that's meroplankton will have will never switch over somehow to a holoplankton. The meroplankton adult is on the bottom, and the juvenile stage is in the pelagic zone. And you should know some examples. You should be able to give some examples of meroplankton. And some of these I think you should know already. Crabs, mollusks, barnacles, all have planktonic juvenile stages. And then you should know some of the important holozooplankton. We're going to talk more about Proteus on Thursday and next week. These are names that you should know: copepods, euphausids, salps, and Cnidarians. They all have different roles in the plankton. All have different. They eat different organisms, and for that reason, they have different roles. So that's a kind of a start on a very fascinating group of animals for the most part. When we talk about Proteus, we won't be able to use that word animal, but certainly for the rest of these organisms, they're really important and representatives of the animal kingdom that we see in the marine world.